ontologically. And then I narrowed down my search even more by focusing on those genes that were involved in cell adhesion, motility, and migration. And from those genes, I identified MAC kinase phosphatase 3, or MKP3, which was consistently upregulated by wild-type p63, but not by the mutant derivatives, both at the mRNA level as well as at the protein level. Okay. MKP3 is an interesting target because it is a negative regulator of ERK signaling. Under basal conditions, MKP3 keeps low levels of phosphorylated ERK by dephosphorylating both the threonine and the tyrosine residues found on the activation loop of ERK1 and ERK2 on the TEY motif. But when ERK is activated by growth factors or mitogens via the RAS-RAF-MEK cascade, ERK can direct the very rapid degradation of MKP3 protein via the proteasome pathway, and then it can go on and exert these biological functions, which include cell growth, proliferation, survival, differentiation, motility, and metabolism. However, ERK2 also upregulates MKP3 at the transcriptional level. So then MKP3 can accumulate in cytoplasm and act as a negative feedback inhibitor in order to restrain signal strength and duration. And MKP3 was particularly interesting to us because ERK can promote the metastatic process in a variety of ways. It can promote focal adhesion turnover, which is a process that is required for cell migration. It can directly phosphorylate myosin light chain kinase and thereby promote actin myosin contraction. And it can translocate into the nucleus where it can upregulate matrix metalloprotease expression, thereby promoting extracellular matrix degradation and cell invasion. And very importantly, ERK signaling is well known to be upregulated in invasive cases of breast cancer. So I decided to look at the expression of p63 and MKP3 in invasive breast cancers. And I used Oncomine to do this, and I found that both p63 and MKP3 have decreased expression in invasive breast carcinomas, right? So this was very encouraging. And we asked the question, well, then can delta MP63 alpha affect cancer cell invasion via MKP3? Is MKP3 important for mediating these effects? Okay. So to answer this question, for now I had to overexpress p63 in the highly invasive and tumorigenic breast cancer cell line MDA MB231. As expected, p63 upregulated MKP3 at the mRNA level as well as at the protein level. But surprisingly, it did not significantly alter phosphorylated ERK levels. So we scratched our heads for a little while. And then we reasoned that this is probably because MDA MB231 cells express hyperactivated RAS and RAF proteins, thus likely masking the effect of upregulated MKP3 on ERK phosphorylation. However, it has been reported that MKP3 can inhibit ERK nuclear activity by sequestering ERK1 and ERK2 on the cytoplasm. So I wonder whether this could be the case in our system. So I generated a mixed population of p63 positive and p63 negative cells, and then assessed phosphorylated ERK levels by immunofluorescence. And I found that in a significant number of cells that were p63 negative, the nucleus in that cell expressed higher levels of phosphorylated ERK compared to the cytoplasm, while in the majority of the p63 positive cells, there was a uniform distribution of phosphorylated ERK between the cytoplasm and the nucleus. So when I quantitated this, I found that almost 40% of p63 negative cells had high levels of nuclear phosphorylated ERK, while less than 15% of the p63 positive cells had higher levels of phosphorylated ERK in the nucleus. And in accordance with this, the expression of MMP1 and MMP9, two well-established downstream transcriptional targets of ERK, had decreased expression when we overexpressed delta and p63 alpha. This further confirming that p63 can negatively modulate ERK signaling in this system. So we were satisfied and decided to move on onto the biology again. And we saw, as expected, that p63 overexpression dramatically decreases cell invasion, but the question still remains. Is MKP3 important for mediating these effects? So to test this, I knocked down MKP3 expression this time in a background of p63 overexpression. And I found, and then I subjected these cells to cell invasion assays. And I found, as expected, p63 in the presence of a controlled short herping RNA markedly decreases cell invasion, but when we get rid of MKP3, cell invasion triples. And sure, it doesn't go all the way back up to 100%, but it triples compared to the controlled short herping, indicating that MKP3 upregulation by p63 is at least partially responsible for mediating these effects. So we were fairly happy with our exogenous system. We had found that exogenous delta and p63 can negatively modulate ERK signaling, and that delta and p63 alpha inhibits cancer cell invasion. So we wonder, what about endogenous p63? Can endogenous delta and p63 alpha regulate MKP3 and ERK signaling? So this time, I knocked down p63 in MCF10A mammary epithelial cells. These are non-transformed cells, and in FADU head and neck squamous cell carcinoma cells. And I found that in both cases, when I ablate p63 expression, MKP3 expression is markedly decreased both at the mRNA level as well as at the protein level. So you can see here and there. And importantly, this downregulation of MKP3 expression occurred concomitantly with an increase in phosphorylated ERK levels. However, we did not detect any significant changes on MEK phosphorylation. MEK is the immediate upstream kinase for ERK, meaning that any changes that we find on ERK phosphorylation are not due to increased kinase activity, and therefore must be due to differences in the kinetics of dephosphorylation. And just one more point that I wanted to make is that MCF10A cells express wild-type p53, while FADU cells express DNA binding deficient mutant p53. So we believe that these effects that we see here occur independently of p53 status on the cell. We then wonder whether this regulation was direct or indirect. So I looked at the MKP3 gene and promoter, and I found a putative p63 binding site in the region upstream of the translational start site. And I performed chromatin immunoprecipitation with a p63 antibody, followed by PCR blanking this site. And I found that indeed p63 binds to this region, indicating that the regulation is likely to be a direct regulation on transcription. We looked further into cell signaling, and we found that in both MCF10A and in FADU cells, when we knock down p63, there's an increase in MMP1 and MMP9 expression in a manner analogous to what we found when we overexpressed p63 and MMPs were downregulated, thus strongly demonstrating that p63 can negatively modulate ERK signaling. So we were happy. We're very happy with these endogenous results. So we asked, does reduced delta and p63 alpha affect motility, right? We need to go back into the biology. So I subjected cells that with ablated p63 expression to cell migration and cell invasion assays. And I found that both migration and invasion in both MCF10A and FADU cells was markedly increased when I get rid of p63, right? And not only is migration and invasion increased when you get rid of p63, but p63 ablation promotes focal adhesion turnover. So if you look at the control cells, you'll see how focal adhesions form these large complexes organized around the periphery of